In this video, we are going to have an introduction about biostatistics. This text introduces the students of biology to the concepts and methods used in the biostatistical treatment of data. Broadly speaking, the application of statistical methods to biological problems is called biostatistics. The biological data which results from the conduct of experiments of various kinds are usually collection of measurements and observations. The biological data which results from the conduct of experiments of various kinds are usually collection of experiments, measurements and observations. Broadly speaking, the application of statistical methods to biological problems is called biostatistics. The biological data which results from the conduct of experiments of various kinds are usually collection of measurements and observations. The biological data which results from the conduct of experiments of various kinds are usually collection of measurements and observations. At times, such collections may be large and require classification, summary description, and rules of evidence for drawing valid inferences. The biological data which results from the conduct of experiments of various kinds are usually collection of measurements and observations. At times, such collections may be large and require classification, summary description, and rules of evidence for drawing valid inferences. The biological data which result from the conduct of experiments of various kinds are usually collection of measurements and observations. At times, such collections may be large and require classification, summary description, and rules of evidence for drawing valid inferences. Biostatistics provides a conceptual framework and practical methodology whereby this can be done. Statistical knowledge plays an important role in many aspects of science and education. Some knowledge of statistics is essential for the students of biology, zoology, medicine, psychology, microbiology, and many other disciplines for the reasons given below. First one is the comprehension and use of scientific knowledge. With the proliferation of scientific knowledge and understanding of the literature in many fields of inquiry requires familiarity with statistical methods and modes of thought. Many books and journal articles not only report experimental findings in statistical form but also present theories and arguments involving statistical concepts. The statistical knowledge helps to evaluate the results and significance. One may come across a research article with the results projected as quite significant, but when examined statistically, the results may not be significant at all. A person with the knowledge of statistics often evaluates the scientific results and claims by examining the sample size, that is, the number of individuals or items studied, sample collection methods, and analysis procedures. Understanding of statistics helps to attain an attitude to respect the possibilities of statistical proof with a healthy skepticism. Understanding of statistics helps to attain an attitude to respect the possibilities of statistical proof with a healthy skepticism. In most of biological and medical experiments, the statistics is extensively used to study variability among individuals and effectiveness of different drugs and treatments. A person with the knowledge of statistics often evaluates the scientific results and claims by examining the sample size, that is, the number of individuals or items studied, sample collection methods, and analysis procedures. Understanding of statistics helps to attain an attitude to respect the possibilities of statistical proof within a healthy skepticism. In most of biological and medical experiments, the statistics is extensively used to study variability among individuals and effectiveness of different drugs and treatments. A little understanding of statistics can ensure and enable us to appreciate the role of these approaches in different fields. And familiarity with statistical jargon can result in complete lack of understanding of simple publications. Statistics is often misused or misinterpreted, but to those who understand the misinterpretations, they tell the truth or they say nothing. So that was the first point. Okay, why statistical knowledge is important? It can be used for the comprehension and use of scientific knowledge. Now, the next point, design and conduct of experiments. First one was the comprehension and use of scientific knowledge. How to comprehend and use the scientific knowledge by using biostatistics. The next point is about design and conduct of experiments. How to design some experiments and how to conduct some experiments with the help of biostatistics. Many students, as part of their training, are required to design and conduct original investigations. The design of an experiment is inseparable from the treatment of results and conclusions drawn. A simple experiment, for example, must be designed to facilitate the treatment of data in such a way as to permit clear interpretation. The sample size, sampling method, summarization, and treatment of data must be done in correct scientific way as to withstand the evaluation of experts. The experiments designed and conducted by using statistics often lead to interpretation that conforms to non-scientific evidence. If the design is seriously flawed, no amount of statistical manipulation can lead to the drawing of valid conclusion. So it can be used in theoretical research. The research in the field of animal behavior has become more quantitative. The development of theories serve to organize our information. Theorists predict that we expect to observe in specified circumstances. The means by which we test the theories are largely statistical. Next is the problem solving type. Problem solving type. Often research is conducted on a limited scale. Research is conducted on a limited scale, not to test a theory, but to uncover information vital to the solution of a practical problem. Often, research is conducted on a limited scale, not to test a theory, but to uncover information vital to the solution of a practical problem. We may be interested to seek the answer of such questions. How can a particular test be improved? Is this variable, for example, wing length, related to the other variables like age of the bird? Does the new diet has better effect than the common diet? Such questions call for the application of statistical methods to the available data. So we can use this in the problem solving sector also. Next is daily use. Statistics are of immediate and practical utility. They help us get our work done more quickly and effectively. They aid the teachers in the assignment of grades and the construction of tests. The students can make their results more presentable by using statistical methods. The validity of various types of claims in these advertisements we see daily can be tested by just examining their experimental design and procedures adapted to draw inferences. A person with the basic knowledge of statistics may not find it easier to accept the false claims. A person with the basic knowledge of statistics may not find it easier to accept the false claims. So let's have a definition about biostatistics. Before moving on to that, let's recall what are the major points learned. So, what is the importance of biostatistics, which can be used for the comprehension and use of scientific knowledge, along with the design and conduct of experiments, then the theoretical research field and problem solving, and along with the daily use. So, these are the major important uses of biostatistics, how we can link the biostatistics with our daily day-to-day life. We can have a daily usage of statistics, problem solving, theoretical research in case of animal biology like ethology, and design and conduct of experiments along with the comprehension and use of scientific knowledge. So these are the major points. Then let's have a definition of biostatistics. Biostatistics deals with the application of statistics to the biological data. Biostatistics deals with the application of statistics to the biological data. By statistics, we mean the aggregates affected to a marked extent by multiplicity of causes numerically expressed, enumerated or estimated according to reasonable standard of accuracy, collected in a systematic manner for predetermined purpose, and placed in relation to each other. It involves collection, arrangements, analysis, and interpretation of the numerical facts so as to draw scientific conclusions or to make effective decisions. It is branch of applied mathematics and may be regarded as mathematics applied to the observational biological data. The students should not try to make rigid lines of demarcation between biology, biostatistics, statistics, and mathematics, as it is not possible and probably also not desired to do so. Biostatistics is simply the application of statistical method to the solution of biological problems. It is also called biometry. There are two types of statistics more commonly used in biological science. Biometry is also another term. There are two types of statistics more commonly used in biological studies. These are simple statistic, simple statistics, and test statistics. Simple statistics and test statistics. Sample statistics and test statistics. So let's learn about what is sample statistics along with test statistics. Okay, so first one is the sample statistics. It is utilized to calculate population parameters. It is utilized to calculate population parameters such as mean, standard deviation, etc. It explains the nature of distribution of data. Data in this case are immediately calculated in order to provide biologists the best hand results. It also gives the knowledge of statistically significant or non-significant results. It is utilized to calculate population parameters such as mean, standard deviation, etc. It explains the nature of distribution of data. The data in this case are immediately calculated in order to provide biologists the first-hand results. It also gives the knowledge of statistically significant or non-significant results. So that is what is explained by sample statistics. Next one is the test statistics. It is used to test statistics. It is used to test the hypothesis about one or more sample data. The statistical test one has to choose more data for analysis obtained by conducting the experimental design. It includes the study of chi-square and t-test, etc. Biostatistics can be divided into two subcategories, namely descriptive biostatistic and inferential biostatistics. Which are the two types? Descriptive biostatistics and inferential biostatistics. So, descriptive biostatistics. The study of biostatistical procedures which deal with summarization of data to make it more informative and comprehensible is called descriptive biostatistics. Descriptive biostatistics. The descriptive biostatistics involves The descriptive biostatistics involves graphical and tabular approaches to describe, summarize, and analyze the data. So, the descriptive biostatistics involves graphical and let's continue. So, the descriptive biostatistics involves graphical and tabular approaches to describe, summarize, and analyze the data. This includes anything done to data which is designed to summarize or describe them without attempting to infer anything that goes beyond the data. An example of descriptive biostatistics is the decennial census of India, in which all the citizens are required to provide such information as age, sex, religion, marital status, education, and occupation. The data obtained in such a census can then be compiled and arranged to tables and graphs that describe characteristics of the population at a given time. An example of the descriptive biostatistics is the census of India. So next one was the inferential biostatistics. Although descriptive biostatistics is an important branch of statistics, statistical inference usually arises from samples, and this means that its analysis will require generalizations which go beyond the data. The procedures which serve to make generalizations on the basis of studies of a sample, which is a part of the population of interest, constitute inferential biostatistics. Such methods are required, for instance, to predict the mean weight of fishes in a pond. A big pond, to compare the effectiveness of two reducing diets. Such methods are required, for instance, to predict the mean weight of fishes in a big pond, to compare the effectiveness of two reducing diets on the basis of weight losses of two groups of persons who have been on diet. A scientist studying the weight gains in pigs after being fed on a new diet after taking the weights of control and experimental groups may be interested to find out if there is any real difference in the weights of pigs fed on normal diet and those fed on the new diet. He may also be interested to draw conclusions about the efficacy of new diet and may like to generalize his findings. He may also be interested to draw conclusions about the efficacy of the new diet and may like to generalize its findings. The ability to make generalized conclusions, inferring characteristics of the whole from the characteristics of his parts, lies within the realms of inferential statistics. Statistical inference is the process of drawing conclusions about population characteristics based on the information contained in a sample. So, now let's have a brief history of biostatistics. What is the history of biostatistics? Let's learn about that. Francis Galton in 1822 to 1911, a cousin of Charles Darwin, is called the father of biostatistics and eugenics. Before 19th century, statistics was mathematical, political and governmental. In the early 19th century, a famous Belgian statistician Quiltet combined the theory and practical methods of statistics and applied them to the problems of biology, education, medicine, and sociology. Quitlet is credited with the development of statistical theory as a general method of research applicable to any observational science. Beyond any doubt, the individual who had the greatest effect upon the introduction and use of statistics in biology was Francis Galton, who is also referred to as the father of biostatistics. The inadequacy of Darwin's genetic theories stimulated Galton to try and solve the problems of heredity. In the course of his long life, he made notable contributions in the fields of heredity, eugenics, psychology, anthropometry, and statistics. Our present understanding of correlation, the measure of agreement between two variables, is credited to him. Person collaborated with Galton in later years and was instrumental in developing many of the correlation and regression formulas that are in use today. Galton also contributed to the development of centiles or percentiles. The inadequacy of Darwin's genetic theories stimulated Galton to try and solve the problems of heredity. In the course of his long life, he made notable contributions in the field of heredity, eugenics, psychology, anthropometry, and statistics. Our present understanding of correlation, the measure of agreement between two variables, is credited to him. Person collaborated with Galton in the later years and was instrumental in developing many of the correlation and regression formulas that are in use today. Galton also contributed to the development of centiles or percentiles. The term biometry was coined by W.F.R. Weldon in 1860 to 1906. In the 20th century, new techniques and methods were applied to the study of small samples. The major contributions in small sample theory were made by an English statistician, P. A. Fisher. He developed most of his methods in an agricultural and biological setting. Fisher's methods are used in almost all the fields of science. Statistics is a broad and extensively active field whose applications touch almost every aspect of science. Although statistics arose to satisfy the needs of scientific research, the development of its methodology in turn affected the sciences in which statistics are applied. Now let's learn about some new topic. We are going to answer a question. What can be done with statistics? We are trying to answer that question. We are trying to answer that question of what can be done with biostatistics. Biologists mainly deal with animals in which variability is much more as compared to the other systems. A knowledge of biostatistics aids in the correct interpretation of data from animal systems. Suppose a biologist wants to carry out an experiment to study the effect of a new diet on the weights of rats. He can take the help of biostatistics to decide how many rats should be included in the experiment, how to select normal and experimental rats, to make observations more concise and informative, and to draw correct conclusions. What are some of the things that can be done to get the maximum information from the results of such experiments? Here is the list of some of the possibilities. Average can be calculated. These averages give a picture of the typical performance of the groups. The variability of the measurements can be determined. Averages can be calculated with that information. These averages give a picture of the typical performance of the groups. The variability of the measurements can be determined by using the average as a point of reference. One can determine how the observations spread about its central point. We can easily calculate the average. The variability of the measurements can be determined by using the average as a point of reference. One can determine how the observations spread about its central point. Graphs, tables, and figures can be prepared to portray clearly the nature of the group or groups. The raw observations can be transformed into a more meaningful form. The most common of these forms are centiles or percentiles and standard scores. Firstly, we can calculate the average. Next, we can learn about the variability. Third one, we can present the data in the form of graphs, tables, and figures. The raw observations can be transformed into a more meaningful form. The most common of these forms are centiles or percentiles. The relationship of one variable to another can be determined. The statistics called correlation coefficients are among the most useful. For example, it might be of interest to find the relationship between the height and weight, between the dosage and the response. The reliability of the measurement instruments can be determined. This is done by making two measurements of the same individual with the same or parallel devices and finding the correlation between the two sets of data. As will be said, there are also other ways of computing reliability coefficients. The reliability of the measurement instruments can be determined. This is done by making two measurements of the same individuals with the same or parallel device and finding the correlation between the two sets of data. As will be seen, there are also other ways of computing reliability coefficients. The variability or the validity. The validity of the measurements can be determined. The reliability of the measurement instruments can be determined. The validity of the measurement can be determined. For example, intelligence tests are often validated by correlating scores on these tests with guide point averages. If the intelligence tests are valid, those who obtain the highest scores will also receive the highest academic grades. So, here we are conducting a validity test. One set of measurements or a combination of variables can be used to predict future status or behavior. This is probably the major end of all correlational work, and themselves correlation coefficients are of little value. One set of measurements or a combination of variables can be used to predict future status or behavior. This is probably the major end of all correlated work. In themselves, correlation coefficients are of little value. When they are combined with other information, they are of great value, but when they are used alone, they are of no value. From the measurement of a they are of a lit, they are of with little value. From the measurement of a sample of individuals, inferences can be made about the larger population from which the samples are drawn. The drawing of statistical inference is one of the chief activities of modern research. Information about a small sample is usually of limited use. The research worker hopes to generalize and draw conclusions about a larger group. The performance of one group can be compared with that made by another, and the significance of any difference can be tested. Suppose that a school is trying out a new reading curriculum. We have two groups. One is handled in the usual way in the course of a semester or a year, and on the other, the performance of one group can be compared with that made by another, and the significance of any difference can be tested. Suppose that a school is trying out a new reading curriculum. We have two groups. One is handled in the usual way in the course of a semester or a year, and the other, an experimental group, is treated in a new fashion. That is, the experimental group is exposed to new reading curriculum. At the end of the school year, the same reading test is administered to both groups. The scores made by each group on this test can be averaged. By inspection, it cannot be determined whether or not the averages are significantly different. At this point, a test of significance can be made to see whether or not the difference is attributable to change variable. The process of drawing inferences, making predictions, and testing significance are examples of sampling or inferential statistics. The other methods mentioned are included in what is called descriptive statistics. Modern experimental research in fields of knowledge from anthropology to zoology uses sampling statistics. Modern experimental research in fields of knowledge from anthropology to zoology uses sampling statistics. Now let's learn some definitions and concepts about statistics. First, let's learn about what is data. While observing various phenomena, one is usually interested in getting information on specific characteristics, example age, sex, weight, height, marital status, occupation, blood pressure, body temperature, etc. These characteristics are referred to as variables, whereas the values of observations recorded from them are referred to as data. Thus, the data are the raw materials for statistics. There are two types of statistical data. Primary data. What is primary data? It is the data collected by a particular person or organization for his or her own use from the primary uses. It is the data collected by a particular person or organization for his or her own use from a primary uses. Secondary data. It is the data collected by some other person or organization for his or her own use, but the investigator can also use it. It means that a data can be primary for one person and secondary for the other. Now let's learn about what is population. A population is any actual or conceptual collection of individuals defined by stated characteristics. Every set of statistical data must be interpreted in relation to the population. We were learning about the population. A population is any actual or conceptual collection of individuals defined as stated characteristics. Every set of statistical data must be interpreted in relation to the population. Population is a group of individuals about which the scientific interests are made. All university students in Delhi, all bacteria in a specific bottle of milk, all progeny of a particular rat, all persons who undergo heart surgery in 1998, etc., constitute different populations. On occasions, biostatistician can deal with the population of measurements. By this is meant an indefinitely large aggregates of measurements which hypothetically might be obtained under specific conditions. So, what is a variable? A variable is any property of an individual, person, tree, bird, rat, etc., that can be expressed in numerical terms. A variable is any property of an individual, person, tree, bird, rat, etc., that can be expressed in numerical terms. For example, the height of a 12-year-old boy, the number of leaves on a tree branch, liver weight of a rat, percentage of calcium in the blood of a patient. We are interested in the values of a variable, sometimes of several distinct variables that relate to the different members of a population. A variable may be readily measured or determined for any member of the population. It may be the product of a complicated analysis or laborious counting. Variable is a property whereby the members of a group or set differ from one another. The particular values of a variable are referred to as variable or varied values. A variable may be continuous or discrete. A continuous variable may take any value within a defined range of values. The possible values of the variable belong to continuous series. Between any two values of the variable are indefinitely large number of it in between values may occur. Height, weight, and chronological time are examples of continuous variables. A discrete variable or a discontinuous variable can take specific values only. Size of a family, the number of female patients, the number of leaves, etc. are discrete variables. Variables are of two types. Continuous variable may take any values within a defined range of values. The possible values of a variable belong to continuous series. Between any two values of the variable are indefinitely large number of in-between values may occur. Height, weight, and chronological time are examples of continuous variables. Continuous variables, we can see middle values, in-between values between numbers. A discrete variable or discontinuous variable can take specific values only. Size of a family, the number of female patients, the number of leaves, etc. are discrete variables. When we take the case of a family, number of family members, we can have 2.5 members. We can't have that. We can just have whole numbers. So that is what is meant by the discrete variables. Besides these two categories of measurement variables, which can be expressed in numerically ordered fashion, there are other two types of variables which are often used in biology. These are the ranked variables and attributes. A variable which is expressed as a discrete variable or discontinuous variable can take specific values only. Size of a family, the number of female patients, the number of leaves, etc. are discrete variables. Besides these two categories of measurement variables which can be expressed in numerically ordered fashion, there are other two types of variables which are often used in biology. These are the ranked variables and attributes. Ranked variables and attributes. A variable which is expressed as a series of ranks such as 1, 2, 3, 4, 5 is called ranked variables. For example, in an experiment, one may record the rank order of litters born or of emergence of pupae without specifying the exact time. Variables which are expressed qualitatively are called attributes. These are all properties such as red or white, pregnant or not pregnant, dead or alive, etc. Such are categories are called attributes. Attributes are nothing else but the variables which are expressed qualitatively. We are expressing the quality, and these are some type of variables which we term as attributes. Now let's learn about what is a constant. The term constant refers to a property whereby the members of a group do not differ from one another. So what is a constant? The term constant refers to a property whereby the members of a group do not differ from one another. In a sense, constant is a particular type of variable. It is a variable which does not vary from one member of a group from another or within a particular set of defined conditions. Suppose we are interested in religious attitude among students in a college. Of prime concern is the variable of religious attitude. However, other variables such as sex, age, may have to be taken into account in interpreting our findings. The membership of a specific college and the time when data are collected are constants. Sample. A sample is any subgroup or sub-aggregate drawn by some appropriate method from a population. It is some segment of the population that is examined and measured as an aid to investigating properties of the parent population. As a sample of college students, One might take all the students of first year zoology class in a college. Similarly, 2 ml milk from a bottle of milk taken for bacteriological studies is a sample. Now, what is a representativeness and randomness? Though any part of a population can be regarded as a sample, for the sample to be useful in making inferences about the population, it must be some sense be representative. In some respects, the first year zoology students might prove representative at least of of the students' body of other colleges in height and weight, for example, but not in age or in knowledge of modern history. 2 ml milk will not be representative bacterial population unless one ensures that the bottle is well mixed before pouring. The only way of ensuring that a sample is representative is to introduce an element of randomness into its selection. After the population has been exactly defined, the sample is drawn by lots. Sample methods are discussed later. The only way of ensuring that a sample is representative is to introduce an element of randomness into its selection. After the population has been exactly defined, the sample is drawn by lots. Sampling methods are discussed later. So, what is parameters and statistics? A parameter is any numerical property descriptive of a population. Population mean, median, and mode are the parameters of location. They relate to the general location of numerical values of a variable. A statistic is any numerical property calculated for a sample. The statistics one calculates varies from sample to sample. Often a correspondence exists between sample statistics and population parameters, and inferences about an unknown parameter may be based on calculations with the non-sample statistic. Mean height of students of a college is parameter, and mean height of students of first year zoology students are the of the college is a statistic. What is a statistic? A statistic is any numerical property calculated for a sample. A statistic one calculates varies from sample to sample. Often a correspondence exists between sample statistics and population parameters, and inferences about an unknown parameter may be based on calculations with the non-sample statistic. Now let's learn about accuracy and precision. Accuracy and precision are used synonymously in everyday speech, but in statistics we define them more rigorously. Accuracy is the closeness of a measured or computed value to its true value. Precision is the closeness of repeated measurements of the same same quantity. A biased but sensitive scale might yield inaccurate but precise weight. By chance, an insensitive scale might result in an accurate reading, which would, however, be imprecise since a repeated weighing would be unlikely to yield an equally accurate weight. Unless there is bias in a measuring instrument, precision will lead to accuracy. We need, therefore, mainly be concerned with the former. Precise varieties are usually, but not necessarily, whole numbers. Thus, when we count 4 eggs in a nest, there is no doubt about the exact number of eggs in the nest if we have counted correctly. It is 4, not 3, not 5, and clearly it could be 4 plus or minus a functional part. Meristatic variables are generally measured as exact numbers. Seemingly continuous variables derived from Meriastic ones can, under certain conditions, also be exact numbers. For instance, ratios between exact numbers are themselves also exact. If in a colony of rats there are 18 females and 12 males, and the ratio of females to males is 1 is to 5, continuous variable but also an exact number. For example, if we report that the wing length of a bird is 8 cm long, we are stating the number 8 as an estimate of the true wing length. For example, if we report that the wing length of a bird is 8 cm long, we are stating the number 8, a value of continuous variable, as an estimate of the true wing length. This estimate was made using some sort of measuring device. Had the device been capable of more accuracy, we might have concluded that the wing was 8.32 cm long. When recording values of continuous variables, it is important to designate the accuracy with which the measurement has been made. By convention, the value 8 denotes a measurement in the range of 7.5 to 8.4999, and the values of 8.3 designates a range of 8.2500 to 8.34999, and the value of 8.32 implies that the value lies within the range of 8.31500 to 8.324099. That is, the reported value is the midpoint of the implied range, and the size of this range is designated by the last decimal place in the measurement. The value of 8 cm implies a range of accuracy of 1 cm. 8.3 cm implies the range of 0.3 cm, and 8.32 cm implies a range of 0.01 cm. Thus, to record the values of 8.0 implies greater accuracy of measurement, that thus recording a value of 8. For in the first instance, the true value is said to lie between 7.9500 and 8.0499 within a range of 0.1 cm, whereas 8 implies a value between 7.500 and 8.0499 within a range of 0.1 cm. These digits are a number that denote the accuracy of the measurement are referred to as significant figures. This 8 has one significant figure, 8.2 and 8.3 are two significant figure, and 8.00 have three significant figure. In working with exact values of discrete variables, the preceding considerations do not apply. That is, it is sufficient to state that a frog has 4 limbs, or that its left lung contains 13 flux. The use of 4.0 or 13.00 would be inappropriate, for since the numbers involved are exactly 4 and 13, there is no question of accuracy of significant figures. But there are instances where significant figures and implied accuracy come into play with discrete data. An entomologist may report that there are 72,000 moths in a particular forest area. In doing so, it is probably not being claimed that this is the exact number, but is an estimate of the exact number, perhaps accurate to two significant figures. In such a case, 72,000 would imply a range of accuracy of 1,000, so that the true value might lie anywhere between 71,500 to 72,500. If the entomologist wished to convey the fact that this estimate is believed to be accurate to the nearest 100 to three significant figures rather than to the nearest 1,000, he had better present his data in the form of significant notations as follows. If the number 7.2 into 10 raised to 4 about 72,000 is written, a range of accuracy of 0.1 into 10 raised to 4 is implied, and the true value is assumed to lie between 71,500 and 72,500. But if 7.20 into 10 raised to 4 were written, a range of accuracy of 0.01 into 10 raised to 4, that is 100, would be implied, and the true value would be assumed to be in the range of 7,190,950 to 72,050. This accuracy of large values, and this applies to continuous as well as discrete variables, can be expressed successively using scientific notation. Now let's learn about the applications of biostatistics. Biological experiments deal with living things, and the variability among the individuals is quite common. Variations are the basis of evolution of new forms of life. It is quite possible that these variations may mislead the experimenter. He might consider the natural variations as the differences resulting from his treatment, or vice versa. He might consider the natural variations as the differences resulting from his treatment, or vice versa. With the application of statistical methods, such confusions are unlikely to occur. Statistics is used extensively in different branches of biology. In genetics, we often study whether or not the new generations follow the Mendelian ratios. With the use of chi-square tests, which deal with observed and expected frequencies, we can test such hypotheses. The frequencies of different genes and their changes due to the effects of forces like Mutations, migration, isolation, and selection can be estimated with the application of different statistical methods. In ecology, the statistical approaches deal with different aspects of populations and their environment. Ecologists often measure variations in environment and their effects on the different characteristics of organisms. Also, association or lack of it among pairs of groups are of ecological interest. Various procedures like correlation analysis, chi-square, frequency, along with binomial and geometric distributions are frequently used by ecologists. In taxonomy, the statistics is of great help where one has to classify the taxonomic units on the basis of different characteristics and also find out associations or lack of it among various characteristics of groups. With help of statistics, one can draw inferences about the populations. Similarly, statistics is used in almost all the branches of biology like ethology, environmental biology, cell biology, physiology, etc. There are some sort of statistical symbols. There are a number of symbols used in statistics. However, there is no absolute conformity in the use of the symbols and notations. Usage will vary somewhere, and this can be very different. A common and simple statistic location is summation f is equal to n, capital N. Summation f is equal to capital N. This equation is read as capital sigma summation of or the sum of the frequencies is equal to n, the number of individuals. Summation sign is one of the most commonly used statistical symbols. If we are dealing with one variable, a variable is characteristic that manifests differences in magnitude or quantity is designated as x. One measurement or observation of x variable is usually symbolized by corresponding capital letter. Hence, capital X is one measure for variable small x and capital Y is one measure for variable small y. Different individual measures on the same variable are denoted by numeric subscripts. Number of individuals, capital N. Statistic, small n. Mean, mu. Statistic, x bar. Variance, sigma square. Standard deviation, just sigma. Proportion, p. Pearson correlation coefficient, r. Number of cases, small n. And statistic is large n. So, scales of measurement. We conclude this introductory chapter with a note on the types of data which can be subjected to statistical analysis. Data may be classified according to their scales of measurement as follows. Nominal scale. On a nominal scale, numbers are used as labels and no ranking is possible. For example, groups of patients could be labeled 1, 2, 3, corresponding to the classification normal, diabetic, and hypertensive. That is something which is termed as a nominal scale. Next is the ordinal scale. On an ordinal scale, scores can be assigned to the observations in such a way that the rankings obtained are meaningful, but the intervals between the rankings need not be defined. For example, in a survey, people's opinion of a product may be ranked according to the scale poor, fairly good, good, very good, and excellent. That is ordinal scale. Next is the interval scale. In an interval scale, both the ranking and intervals are meaningful, but there is no meaningful zero. An example is the measurement of temperature in degrees Celsius. Next is the ratio scale. On a ratio scale, the measurements may be ranked, the intervals are meaningful, and there is a meaningful zero. Thus, the ratios of measurements are also meaningful if they are made on a ratio scale. Measurements of quantities such as length, area, and time are made on ratio scale, as are counts such as the number of arrives of cars on a service station in a given period of time, which can also take the value of 0. So we are considering the value of 0 also. So that is what is explained by using a ratio scale. So which are the scales of measurement? Nominal scale, ordinal scale, interval scale, and ratio scale.